Hello. Today I will talk about congenital adrenal hyperplasias that are. Um, you have just learned about steroid hormone synthesis, so I would like to start with a little summary of what you learned and how these disorders are developing in lack of the different enzymes that are important in the different steroid hormone synthetic pathways. So, as a recap from the previous block, the starting material for all steroid hormones is cholesterol. Cholesterol can derive from food or our liver can synthesize it. Most enzymes that participate in the steroid hormone synthetic pathway belong to the cytochrome P450 family, coded by the so-called CYP gene family. So this is just a little reminder that you learned about the key enzyme being desmolase, the very first enzyme in the steroid hormone synthetic pathway. And this is already a CYP. And when we go down the steroid hormone synthetic pathway and we branch off to aldosterone, which is a mineralocorticoid, cortisol, which is glucocorticoid, and sex hormone synthesis, different CYP derived enzymes, P450, so-called mixed function oxidases or hydroxylases, play a role in determining which pathway is catalyzed and which hormone is synthesized. We also talked about the localization of these different hormones and hormone family synthetic pathways. So we went through mineralocorticoids are synthesized in a zona glomerulosa. The glucocorticoids are synthesized, of course, from the first couple of steps after cholesterol in the zona fasciculata, and then androgens or testosterone is synthesized in the zona reticularis. Worth to mention that these are prehormones, androgens, and then the final active male hormone is synthesized in the testes, and the estrogen, the female hormone, is synthesized in the ovaries and the placenta. So what are congenital adrenal hyperplasias? This is a collective name for several genetic disorders affecting key enzymes. So there is a mutation in a gene that codes for a key enzyme which catalyzes a step in steroid hormone synthetic pathway. The inheritance pattern of all of these hyperplasias is autosomal recessive. And depending on what enzyme is affected, the consequences are excessive or deficient production of one or the other type of hormone, sex hormones, mineral or glucocorticoids. The most frequent signs, again, depending on which enzyme is affected, salt imbalance due to mineralocorticoids, ambiguous genitalia, extreme virilization of males, sexual infantilism or infertility, mainly due to sex hormone synthetic pathway inefficiencies. So I will go through 4 different examples. Please be aware and please know the enzyme and the gene which is affected and where this particular enzyme is localized in the steroid hormone synthetic pathway, so that logically you can expect what the consequences will be if the gene is mutated, enzyme is not working properly or completely missing from the system. So the first example is the so-called 3-hydroxysteroid dehydrogenase, HSD3B2 gene mutation. This enzyme is missing, and please note that this enzyme is before the branching of the different kinds of steroid hormones. So consequently, because this enzyme is not working, none of the steroid hormones can be synthesized. So there will be a lack in mineralocorticoids, there will be a lack in cortisol or glucocorticoids, and there will be lack of sex hormone, testosterone, estradiol. Increased salt excretion is due to the lack of aldosterone, and female-like genitalia development due to lack of testosterone are hallmarks of this enzyme deficiency. The second example is, this is also less common, congenital adrenal hyperplasia, is the lack of 17-alpha hydroxylase. The gene which is affected is so-called CYP17. Now, CYP17 is localized after the first branch point. What is the consequence of that? That there will not be a problem with synthesizing mineralocorticoids, but there will be a problem with the synthesis of glucocorticoids and sex hormones. So it is not just that there is no problem with mineralocorticoid, but because of this enzyme deficiency, more cholesterol will be used to synthesize aldosterone, which means that there will be an excess, an increased amount of aldosterone circulating in the body, causing sodium retention, fluid retention, thus high blood pressure. However, sex hormones will be decreased, and again, as just before, female-like genitalia will develop in the babies that have this mutation. The third example is the most common of the congenital adrenal hyperplasias, and it is due to 21-alpha hydroxylase deficiency. The gene is CYP21A2. So where is it located? It's located in the aldosterone mineralocorticoid pathway, and it is also located, the same enzyme is working at the beginning of the glucocorticoid synthetic pathway. Thus, insufficient cortisol and insufficient aldosterone production is typical of this disorder. Sex hormone production actually will be higher than normal. So what are the consequences then? Then mineralocorticoids are not synthesized. Salt imbalance is, of course, due to that one, but and cortisol is not synthesized. Now, ACTH, you already learned about it, is a pituitary hormone which regulates cortisol synthesis. So if cortisol cannot be synthesized because of this enzyme deficiency, the ACTH will be actually higher and higher and higher because the body feels that cortisol is not synthesized. So it is typical, one of the hallmarks of 21-alpha hydroxylase deficiency is higher than normal ACTH level, but lack of cortisol. As I mentioned, lack of aldosterone too. However, there will be increased male hormone synthesis, and some of them, of course, will turn into female sex hormones in the female organs. So if there is an increased sex hormone from majorly birth synthesized, the typical sign of this particular hydroxylase deficiency is early virilization of the males and precocious sex organ development in the females. The second most common adrenal hyperplasia is the 11-beta hydroxylase deficiency, coded by CYP11B. So where is this enzyme? This enzyme is a step further down in aldosterone synthetic pathway and a step further down in the cortisol synthetic pathway. So again, insufficient cortisol and aldosterone will be produced. However, there will be an increase in deoxycorticosterone because it is before the enzyme, and it will result in fluid retention. This is already acting as a hormone. Early virilization again is typical as in the previous example. Since these two pathways are blocked, more male hormone will be synthesized. So early virilization, precocious sex organ development in females are typical of this particular congenital adrenal hyperplasia subtype. So, very quickly, what I talked about today is we recapped very quickly that for steroid hormone synthesis, we need the cytochrome P450 system, which is called mixed function oxidase, hydroxylation, and monooxygenation reactions are catalyzed. Numerous cytochrome P450s participate in steroid hormone synthesis. And steroid hormone synthesis is located in the adrenal cortex. Corticosteroid androgens are synthesized, and further, the sex hormones are active. Sex hormones are synthesized in the testes, ovaries, and placenta, depending on male or female character. Congenital adrenal hyperplasias that were the focus of this very short lecture, the absence of one or more enzymes. These are coded by CYPs, and it usually leads to imbalance in steroid hormone production. Depending on which enzyme is missing, which gene is mutated, and please remember the 4 examples we will see as hallmarks of this disease: the salt imbalance, the ambiguous genitalia, and in some cases, extreme virilization of the males, or in other cases, sexual infantilism, which means not developing normally sexual organs, or sexual organs do develop, but the result is infertility. Thank you for your attention.